All right, so let's look at protein absorption. All right, so as an overview, we know we're consuming proteins in our food and nothing happens to them really other than the chewing, mechanical digestion in the mouth. So once it gets to the stomach, we start chemically digesting proteins into smaller polypeptides. So there's a different color. So into polypeptides. And we do this using pepsin. Okay. Then the proteins enter the small intestine. So small intestine. And this is where we get small peptides. So small peptides. And we get that through the use of pancreatic enzymes. And those enzymes include trypsin, chymotrypsin, and carboxypeptidase. And peptidase. So those are the three protein or proteases that are there to break down proteins into smaller peptides, so small peptides. All right, so once we have that, the small peptides, then in the small intestine, still in the small intestine, these small peptides come into contact with the brush border. And at the brush border, these small peptides are turned into tripeptides, dipeptides, and amino acids. So tripeptides, so 3 amino acids there, 2 linked, or just amino acids. Okay, so looking at our cell. So let's look at our cells here. So make a big cell here, big cell here, and our little blood vessel here. Okay, so we know this right here, all the way down, is the brush border. So once we have small peptides that get like that, once the chyme enters the duodenum, we have polypeptides that are smaller than, or broken down a little bit from the stomach, goes in there, and then through these enzymes, trypsin, chymotrypsin, and carboxypeptidase, we get the small peptides. Then those peptides then come to the brush border, and we get tripeptides, and dipeptides, and we also get individual amino acids. Okay, so dipeptides and tripeptides, these two here, they enter the cell with the help of hydrogen. So hydrogen, I'll make this a little bit of a different color. So we've got hydrogen out here in the lumen, and on the plasma membrane we have a transporter that co-transports hydrogen and tripeptides and dipeptides. All right. Also on this plasma membrane is sort of a pump that allows for hydrogen to be shuttled back out into the lumen with sodium coming in. Okay, so we can always have a gradient for hydrogen to bring in those tripeptides and dipeptides through this little antiport thing. Okay, so this is, yeah, it's co-transport. So the sodium potassium co-transport maintains a gradient for hydrogen. So more hydrogen then is out in the lumen than in here, so that this can occur to carry tripeptides and dipeptides into the cell. All right, amino acids come in using sodium. So we've got sodium out here because this is the outside of the cell, so of course there's sodium out there. And put our little transporter thing here, and sodium will come into the cell and, sorry, forget what color I did my amino acid, and amino acids will come with it here. Okay. So And of course, sodium is still going back out through this little co-transport thing here. All right, so in order to keep sodium coming in, of course, we have our friend here, this sodium-potassium pump. Okay, so we're getting sodium that's coming in and coming out as well. Okay, and then of course we have potassium coming in here. All right, and then of course we have another transporter, make that a dark blue, on that's the basement membrane side of the cell, and that allows for the transfer of dipeptides and tripeptides and amino acids into the blood. So now we have all that in the blood headed towards the liver. So that's the tripeptides, dipeptides, and amino acids headed to the liver. Okay, so very similar to the carbohydrate absorption. All right. Okay, so the next one we will look at is fat absorption, or absorption of lipids.